For biology students studying the diversity of life on Earth, why would one study viruses? Now, what is tempting is to say that viruses are not technically alive, and so perhaps biologists should skip that chapter and go on to other things. Now, that's certainly a temptation. I would like to study viruses, as do many biologists, for a large number of reasons. One is that there's so many of them. So if you simply take a milliliter of water from a wetland, this study came from Maryland in the United States, a milliliter could have 4 billion viruses. All right, now viruses are very small, but notice to what degree they outnumber bacteria, which outnumber all other living things. Is Maryland's water overly full of virus? No. So if you were to go to Antarctica, if you were to go to ocean water, if you were to go to volcanic springs, there are just viruses everywhere. And so this is, a major component of ecosystems, just in the terms of number. Now, the term viral ecology is an odd term because when we think of ecology, we often think of living things, producers, predators, etc., food chains, the cycling of nutrients. So why would viruses be involved in the term ecology, where a number of biologists do study the impact of viruses in ecosystems? They have many. So, for example, there are viruses, as I'll mention, which kill bacteria, cause them to burst. When they do, this would then liberate all of the materials inside the bacteria. Their carbohydrates, their lipids, their nitrogen molecules. Now, as I had alluded to in that video, prokaryotes can fix nitrogen, many of them, convert nitrogen gas into usable forms like ammonium nitrates that then get incorporated into DNA and protein. And so other organisms typically can't do that. Animals certainly can't. And so animals need to get nitrogen. The only way that they can is by ingesting it. And so if viruses are causing bacteria to burst, then this causes all of their nutrients to now become available where they can be used by other organisms. So when we talk about food chains and when we talk about the cycling of nutrients, of carbon, of nitrogen, of sulfur, and other things, viruses certainly have a role in causing the lysis of bacteria. So viruses certainly have roles in ecology. Viruses, as I'll mention presently, can actually transfer, not only can they infect bacteria, but every now and then, occasionally they can introduce new genes into bacteria. And so then, as far as the evolution of bacteria, viruses have a role in giving them some genes. Now, I know that sounds very, odd or obscure, but in the study of the human genome, it was determined that about 1% of human DNA was not originally human, but rather viruses whose sequences, after infecting human ancestors, going back to the previous ancestors, early amniotes, early reptiles, etc. This then became part of the genome. And so part of our genome originated not in animals or mammals or primates, but rather in viruses which infected them. And so, viruses can serve as part of a source of evolutionary material. Finally, viruses are food. They have protein, they have DNA, and so therefore they can be ingested and digested for nutrients. And so there are microscopic organisms which can, say, filter feed. That would include a number of protists. Here's a microscopic animal known as a rotifer, but lots of animals filter say seawater or fresh water. So here's a sponge. it makes its living by trapping particles from seawater. Some of those particles are undoubtedly viruses. So viruses are helping to feed animals. Even oddly, our throats catch mucus, or produce mucus, which then catch debris, and we sweep it away from our lungs up into our throat, where we can swallow it into our stomachs. We do that not really as a way of feeding, but rather as a way of protecting our lungs. But nevertheless, viruses in the air end up in this mucus. It ends up in our stomach. We break down the protein nucleic acid. So some of our energy actually comes from viruses. So once again, to treat viruses as, oh, they are not alive, therefore we won't discuss them in a biological setting, neglects the roles that they have in ecosystems. Now, obviously, we focus a great deal on the viruses which infect humans, but viruses can infect virtually everything that has been studied on Earth. So there are viruses which infect plants, there are viruses which infect fungi and cyanobacteria, etc. I'll go into this in a little more detail in subsequent videos, but we could actually give them names if we wanted. So viruses which infect fungi could be called mycophage or mycophage, depending on how you like to pronounce it. Then there can be viruses which are infecting microscopic organisms. So protists can get viral infections, including amoeba can get infected by some of the largest viruses, like the Mimi virus, which I'll mention. So viruses are something which infect lots of different organisms. So viruses can infect bacteria. Viruses can infect not only the eubacteria, but also the archaea, that other group of bacteria that often lives in extreme environments. Viruses can infect the blue-green algae, the cyanobacteria. Now, this has lots of consequences. Once again, it can recycle all of the nutrients, but also something that's interesting, the viruses can actually inhabit these extreme environments where archaea might be found, very high temperatures, etc. In fact, the viral titers there can be extremely high. Some of the highest concentration of viruses in the natural world can be in these extreme environments. So the fascinating thing is, since archaea living in these extreme environments lead us to believe that the earliest life living on the early Earth with many more extreme environments may have been adapted to high temperatures, high acidic environments, etc. And so, when we glimpse bacteria and archaea in these environments, that's perhaps a glimpse of what the earliest life was like. The fact that viruses are so abundant there, once again, perhaps gives us a glimpse of, going back in time, because we think that before there were cells, that life would have started off with simpler things which were RNA based. The only things which use RNA as their genome today are viruses. And so while some viruses continually pop up as new things, it is thought that many viruses are old, once again, perhaps dating back to a pre-cellular world. And not to get into the genetics of it, but there would even be reasons, if you ask, how do diverse viruses, say, make certain DNA nucleotides like thymine? Well, if viruses were simply parasites stealing from their host, then it would be, easy to conclude any mechanisms that they stole from a living host. Well, it turns out viruses have, some do, unique metabolic pathways to make biomolecules that no living host that we know of has. So if you could steal your host's way of making a DNA nucleotide, why would this simple parasite then make up its own? Well, once again, if some of these are dating from a pre-cellular world, there may be clues about, the origin of genetic codes, for example, which comes from the study of viruses. But viruses can infect blue-green algae, the ARCHAEA, and then also eubacteria, and we commonly refer to them as simply bacteriophage or bacteriophage. And so there are a number of different ways that the viruses can infect bacteria, but that'll be part of videos coming up in just a second. So once again, just viruses are specific. The viruses which infect bacteria, often they need, this protein capsid which can actually inject DNA, because bacteria can have cell walls. So to introduce the nucleic molecule of a virus into the bacterial cell, it needs some sort of apparatus to do that. These are not viruses which can infect humans, and so we find specificity in these viruses because the virus capsid has to bind to certain receptors. There are viruses which are larger than we thought viruses could be. And so therefore, these are relatively newly discovered simply because people weren't looking for viruses which were this big, like the Mimi virus. In fact, it is so big that Mimi virus has more genes than the simplest living bacteria. And so it's hard then to say that, oh, let's not study virus because they're not alive, if actually some have more genes than things that we do say that are alive. How can you say that this is something completely in the non-living world, but yet it has such a complex genetic code? Some biologists would take an alternate route and say that, let's say that these viruses are not alive, but now we have a gray area, and some of the larger viruses are. This Mimi virus infects amoeba and depends on this spike protein to interact with receptors. But interestingly, there's a small virus which can then bind these spike proteins and inhibit the Mimi virus's ability to infect amoeba. So in other words, this is a virus which in essence gets a virus. This smaller virus impedes the larger virus's ability to infect cells. So when I said that everything gets a virus, you could even, in that example, make an argument that viruses can actually then affect viruses. All right, so once again, then just giving a little more detail about that Mimi virus. It is part of a group known as the nucleocytoplasmic large DNA viruses. And so they can have a much larger size, which is why it took them so long to be discovered, because some of the filters which were used to studying the viruses would then separate them out, because it was thought that all viruses were smaller than they were. And so as you can see, if this is 3/4 of a micron, there are ARCHAEA, which are under half a micron, or eubacteria, 0.2 to 0.3 microns. So they are larger than some bacteria. And if you look at the size of their genome, 1.2 megabase pairs, here's a cell which is under a half a megabase pair, or 0.6, etc. So they have less DNA than some living cells, with more than 980 genes. Here's a bacterium with 400 genes, with 500 genes. So once again, these are large viruses, larger than some bacteria, more DNA than some bacteria, more genes than some bacteria. So viruses are certainly more diverse than we often give them credit for. The reason that viruses only infect certain things is that the virus has to interact with a specific type of receptor to gain entry to the cell. And so therefore there's a specificity involved, which is why there are certain viruses which only infect fungi or only infect cyanobacteria, once again, since bacteria have cell walls, the bacteriophage, which then infects bacteria, often needs a mechanism which then injects its nucleic acid through the cell wall. But this is why viruses are so specific. Viruses use a nucleic acid, but which nucleic acid they use varies. Some use single-stranded RNA, some use double-stranded RNA. So once again, this is interesting because we think that life began in an RNA world, and viruses are the only things today whose entire genome can be RNA. They can also use single-stranded DNA and double-stranded DNA. Now, during infection, say something with single-stranded DNA may then have to, inside the cell, add new DNA to make it double-stranded in order for it to function in the infection route. Those that have RNA may have to convert that to DNA. And so, for example, in HIV is an RNA virus that must convert itself into DNA. And some of the medications in the multi-drug cocktail, which treat AIDS, that's how they function, in that they can block the reverse transcription of RNA into DNA. Some viruses are called lytic viruses because when they inject their nucleic acid into a cell after binding to the receptor, then they will ultimately cause the cell to burst. Now, what typically happens is once the nucleic acid is injected, the virus's first action is to replicate its nucleic acid, making lots of copies of that. Very often the host genome is broken down in the process. So it replicates its nucleic acids, and then the genes which replicate the viral nucleic acid, they are expressed first. And then later in the infection cycle, the genes which make a protein capsid. So most viruses have only two parts, a nucleic acid and then a protein capsid, which will enclose it. The virus then gets packaged inside the protein capsid. And then in a lytic virus, then the host cell bursts, and this then liberates the viral particles to go and infect other cells. There are other viruses known as temperate viruses, or instead of having a lytic life cycle, they go through a lysogenic life cycle, where instead of causing the cell to burst, they could actually incorporate their viral nucleic acid into the host genome. And so that this is not just bacterial DNA anymore, there is viral DNA in it as well. And so the virus is going to be more patient. It's not going to infect and kill the cell right away, but it will actually then just bide its time. So if this bacteria is successful, every time the bacterium divides, it replicates the viral nucleic acid as well. So the success of the bacteria then leads to the success of the virus. Maybe later, if the bacteria is challenged, so life isn't going as well anymore, then the virus might decide to then become lytic to kill the cell and then to burst it and release more particles. There are viruses which do that in humans. So for example, herpes viruses or AIDS, they can actually incorporate their DNA into the human chromosomes and be patient. Let's say I keep getting a cold sore, I keep getting it right here every single time. What's up with that? Well, maybe that there's a herpes virus that's living in a nerve cell, and that every now and then it activates. And it's usually dormant in incorporated into my DNA, but when it activates, the viral particles move down the same axons that they did the last time, say I got a sunburn or whatever, and then they will go here and then cause another infection. HIV, its its DNA can stay in the host for maybe 10 years without causing full-blown AIDS, during which time someone may be spreading the disease, not knowing that they are HIV positive. And then later, this temperate virus can then enter more of a lytic cycle. So that is another way of viral infection. One of the interesting things is bacteria don't reproduce sexually, but there are a couple of ways in which you can actually have genetic exchange between bacteria. One of the ways is actually using viruses as an intermediary. Now, obviously, this isn't the same, but sometimes I compare this to, plants can reproduce sexually, but sometimes they use bees to help. Bees actually carry the pollen from one plant to another. And so, the bees had a role in this. The virus wants to break down the yellow host chromosome, destroy it, and only then replicate its white viral nucleic acid, which will get packaged into the viral capsids. But sometimes by accident, some of the yellow host genes are introduced inside a viral particle. So that was an accident. The virus meant to put infectious viral nucleic acid inside the protein capsid, but instead it put the bacterial genes. So when this viral capsid then infects bacterium number 2, instead of injecting the viral DNA, it will inject genes from bacteria number 1. So once again, this is not sexual reproduction, but actually now bacterium number 2 now gets new genes from bacterium number 1 thanks to a virus injecting it. In what's called generalized transduction, any old part of the host chromosome, any section of this yellow chromosome might accidentally get packaged inside. In what's called specialized transduction, the red area, which is right next to the site of the host genome where the viral DNA inserts itself, that's what can get copied if the virus is sloppy in how it excises itself from the host genome. So considering HIV, a couple of things. One, it only infects certain cells, those cells which have a CD4 receptor. So once again, viruses rely on a very specific interaction between the host cell and the proteins of their capsid. Inside the host cell, the HIV virus, which is an RNA virus, then has to convert its nucleic acid into DNA using reverse transcriptase. In its transcription, usually DNA gets converted to RNA. In reverse transcription, RNA is converted to DNA. Right. Now, when the particle leaves the cell, notice that it is budding. So some viruses, instead of causing the cell to burst and release lots of particles at one time, they will then bud from the cell membrane and then have a little bit of the host cell membrane surrounding them. And so that is both good and bad. And the bad part is it allows the virus to hide. It looks like a little tiny human cell, coated with a little bit of human cell membrane. The good part about this, as far as controlling infection, is that the virus needs that membrane to be infectious. And so, I'm old enough to remember when AIDS first, became widespread in the 80s. People didn't know how it would be spread. And they could I get it from a tabletop? Could I get it from a drinking fountain? Could I get it from a swimming pool, et cetera? That wasn't known. But because the virus, since it's not technically alive, you can't technically kill it. But you can render it to the point where it's no longer capable of causing infection. And anything that will cause this human membrane to rupture, such as a cold temperature, a dry tabletop, chlorine, and disinfectants, et cetera, then can render it non-infectious. So HIV undergoes budding. Some viruses then have a membrane around them. So in addition to the nucleic acid and the protein capsid, they have a membrane as well. Once again, as I had said, the viral RNA can be converted into viral DNA, and this then can insert itself into part of the human chromosome and lay dormant for a while, for years, before becoming active once again. And this has happened. So once again, when we look at the human genome, a large portion, maybe 1% from the Human Genome Project, it was published, that seems to be from viruses which our ancestors caught. Now, there continue to be new viruses discovered. So Ebola is new in my lifetime. HIV, new in my lifetime, at least newly discovered. The Zika virus, which is one of the causes of microcephaly. There are multiple causes of microcephaly. So there are certain genes which could cause a decrease in the size of the skull and brain in a developing fetus. But this virus can actually be spread by a mosquito bite. So the Zika virus can cause devastating and lifelong changes to a fetus. And so new viruses are constantly being discovered and studied. Obviously, the coronavirus needs to be mentioned, and I'll mention it very quickly in just a second, but I intend on making another video just on coronavirus, so I don't want to, so I'm not going to duplicate everything I say. I'll just make a second a second video. One last statement is viruses are very useful to geneticists in trying to understand how genes work, all right? Because, for example, we use fruit flies a lot. Why? Well, fruit flies are easy to keep. They're small, they're cheap to keep, and you can screen a lot of fruit flies and look for rare genetic events. Some geneticists work with bacteria because they're even smaller, they're even cheaper to keep. And then you can screen rarer genetic events. So for example, if something only happens once every million, say, life cycles, you don't want to count 1 million fruit flies, but you can very easily screen 1 million bacteria. Well, let's say you wanted to study an even rarer genetic event. So you want to say study recombination within a single gene. So recombination can occur, but recombination within a single gene, measuring recombination rates, that's an extremely rare event. And so a lot of biologists would then use viruses, because if you just dilute your series out, you can dilute viral, solutions by, a factor of 1 million, by a factor of 1 billion. And so therefore, after that, you can say, oh, look, I see, some new, change here in, the experiment. So for example, if you have bacteria which normally don't get the mutant virus, but you wanted to see how frequently will the mutant virus have a reverse mutation going back to the infectious form, you could then count, and even something which only happens 1 in 1 million viral replications or 1 in 1 billion, you could study that and quantify that and then come up with rates at which these types of mutations occur. And so geneticists can find the study of viruses useful in studying very rare events. Finally, as I said, I'm going to stop here and then make the next video just about coronavirus, given the grave significance that it has. But this is another RNA virus. Just a couple quick thoughts, and I'll repeat these in the next video. It's unusual in that usually RNA viruses have a much higher mutation rate, and so therefore the nucleic acid can't get to be too big. Otherwise, the virus has so many mutations that it just doesn't work anymore. It's no longer infectious. But the coronaviruses, they are unusual in that they actually have a way of fixing mutations, of proofreading and so that they are an RNA virus, but they have a much larger genome than most. If we consider the evolution of living things, so for example, there were once ancestral dogs, and from these ancestral dogs, we could say, well, here's a dog family, and then the dog family, then diversifies, so that then we have, different genera of dogs, etc. Actually, that was. And so we do that with viruses, even though we don't consider them to be alive. They mutate, and one virus then evolves from another. And so we can see that there are orders of viruses. Coronavirus belongs to an order known as Nidovirales. And so there is an order. So once upon a time, there was an ancestral virus, and from its duplications, we have an order. And then from this order, then there are different families, such as the Coronaviridae family, which has evolved. And so just as there are new species which can evolve, there are new strains of virus which can evolve. So we split the family of these coronaviruses into subfamilies, some of which don't infect humans, some of which cause the human cold, but then some of which causes SARS coronavirus 1, SARS coronavirus 2, the COVID-19, one causes MERS, which can be contracted from camels. And so the next video will consider coronavirus. But once again, a lot of biologists study viruses, whether it be for medicine, for viral ecology, as geneticists. So even though viruses don't meet our definition of what is alive, nevertheless, they are appropriate for study in biology.